Welcome to the Deep Dive. Today we're exploring a hidden world, organisms that might surprise you, even if you think you know your biology. We're talking about fungus-like protists.
Mm,
Specifically, we're diving deep into the phylum Plasmodiophoromycota.
right.
Our mission, to unpack what makes these things tick, their traits, their life cycle, their impact. We're using an introductory mycology chapter as our guide, but, making it engaging, not just dense facts.
Absolutely. And it's key to get this up front. People often group them with fungi, but they're really quite distinct. They play these crucial roles, sometimes, well, pretty destructive roles, especially for plants.
So let's start there. Their basic identity. Necrotrophic endoparasites. Okay,
Mhm.
break that down. Inside the host, feeding on dead or dying tissue.
Exactly. They live inside, and they're essentially feeding off the damage they cause or tissue that's already dying within the host. But it's more than just feeding. They are masters of manipulation.
Manipulation, how so?
Well, they actively change the host. They cause hypertrophy. That's where host cells get abnormally huge. And also hyperplasia, where the host cells multiply like crazy, uncontrolled.
Okay, so picture that.
Yeah.
You've got an infected plant, and its roots are just swollen, distorted.
Yeah, think massively enlarged, twisted roots. That's why Plasmodiophora or brassicae causes clubroot in cabbage. The roots look like, well, clubs. Or sometimes like knobbly fingers and toes.
Right, and this leads to stunting. The whole plant just doesn't grow properly.
Precisely. And interestingly, the stunting can be even worse when the plant isn't flowering. The thinking is maybe the parasite suppresses flowering to reduce competition for nutrients, keep the resources for itself.
Huh, clever in a destructive way. And you mentioned their host range is wide.
Oh, incredibly wide. They hit freshwater algae like a vachera, even other fungus-like organisms,
Yeah.
water molds like Saprolinea, Aclea, Pythium, but also, yeah, lots of vascular plants, aquatic ones, land plants. Think cabbage, sugar beets, grasses, potatoes, even things like watercress, nasturtium, and veronica. Basically, wherever their hosts grow, you can potentially find them.
Which connects directly to their economic impact, I imagine.
Big time.
Yeah.
You mentioned Plasmodiophora, brassicae, clubroot. That's a huge deal for anyone growing cabbage, broccoli, canola, that whole family.
And then there's Spongospora subterranea.
Right, that causes powdery scab of potatoes, gives
Mhm.
the potatoes these rough, scabby spots. And there's even a special form, S. subterranea forma specialis nasturtia, that targets watercress, causing crookroot disease.
Nasty stuff.
And it gets worse. They don't just cause disease directly. These organisms can also act as vectors for plant viruses.
Oh, really? So they carry viruses from plant to plant.
Exactly. They transmit them as they infect the host cells. It's like a double whammy for the plant.
Okay, so these things are clearly effective parasites. What are they actually like inside? You said not quite fungi.
Right. So forget typical fungal hyphae. Plasmodiophorids produce these multinucleate, unwalled protoplasts. Think of them as blobs of cytoplasm with multiple nuclei inside, but no rigid cell wall. These are called plasmodia,
Plasmodia.
like slime molds.
Ah, good question, but no, that's a common confusion. These are different. Plasmodiophorid plasmodia are totally internal parasites. They can't move around on their own like slime mold plasmodia, and they can't engulf food particles. No phagocytosis.
So they just sit inside the host cell.
Pretty much. They live entirely within the host cells or hyphae, often just their membrane pressed right up against the host cytoplasm. Very intimate.
And there are different phases. You mentioned primary and secondary plasmodia.
Correct. There's the primary or sporangial plasmodium. Its job is basically replication, leading to the formation of thin-wall zoosporangia, which release more infectious agents.
Then there's the secondary or sporogenic plasmodium. This one is focused on survival. It produces thick-walled resting spores. These spores are tough, often seem to have chitin in their walls, and can survive really harsh conditions, like drying out completely.
The survival stage. Makes sense. Now, you mentioned something really unique about their division, cruciform.
Ah, yes. This is one of their absolute defining features, cruciform nuclear division. It's quite something to visualize.
Okay.
So imagine mitosis, but the spindle forms inside the nucleus. The chromosomes don't line up in the middle like usual. Instead, they form a ring around the nucleolus, which is that dense spot inside the nucleus.
A ring, okay.
Yeah, and when the chromosomes separate, a whole ring of them moves to each pole. But the nucleolus itself doesn't disappear. It persists and stretches out, elongates between the separating rings of chromosomes.
So the whole structure.
If you look at it from the side during this phase, it looks like a cross, hence cruciform. It's really distinctive.
Wow. That is unique. But you're saying not all their divisions are like that.
Correct. That's important. This cruciform type happens during the growth of the plasmodia, but the divisions leading to zoosporanda formation from the primary plasmodium, or resting spores from the secondary plasmodium, and even the divisions during zoospore formation, those lack this cruciform setup.
Interesting. So it's specific to certain stages.
Seems that way. And speaking of zoospores, the motile cells they produce, they're always anteriorly biflagellate.
Meaning 2 flagellas the front?
Yep, 2 whiplash flagella at the front end, and they're usually unequal in length. And you get zoospores produced at 2 different points in the life cycle, looking pretty similar morphologically, but playing slightly different roles.
Okay, let's trace that life cycle. Sounds complicated. Starts with those resting spores in the soil, right? The tough ones.
Exactly. Those resting spores, released when the host plant dies and rots, can just hang out in the soil or water for, well, potentially years, up to 8 years in some cases.
8 years, just waiting.
Waiting for a suitable host root hair to come nearby. When it does, the resting spore germinates and releases a primary zoospore.
The first infectious stage.
Right. This little zoospore swims over, attaches to a root hair, then its flagella stop beating, they pulled back in, and it forms a cyst, basically hunkers down against the host cell wall.
And then it has to get inside. How does that happen?
This is the really wild part. The penetration mechanism. Inside the cyst, it develops this structure, sometimes called the Rohr, which is German for tube. It's like a long cavity.
Okay.
Inside that tube is a pointed, dense rod called the Stachel, German for spike or sting. The Rohr pushes out against the host wall, forming this bulb-like structure that sticks tight, like in a prosorium.
Like a suction cup with a spear inside.
Sort of. Then the Stachel averts it, shoots out from the Rohr, punctures the host cell wall like a needle, and boom. The entire contents of the zoospore, its protoplast, flows through that tiny hole into the host cell.
Whoa! How fast?
Incredibly fast. Reports say as little as 1 second.
1 second. That's amazing.
It is. This tiny amoeba-like protoplast then gets swept away by the host cytoplasm.
And once it's inside the root hair,
it starts dividing using that cruciform mitosis we talked about, grows bigger, and becomes the primary or sporangial plasmodium. And in P. brassicae, the clubroot one, these primary plasmodia might even fuse together.
more complexity. Okay, so this primary plasmodium grows. Then what?
Once it reaches a certain size, it cleaves, basically splits up into segments. Each segment develops into a zoosporangium. These can be single or clumped together, sometimes in organized clusters called sori.
And these zoosporangia make more zoospores.
Exactly. They produce secondary zoospores. These can be released inside the plant to infect adjacent cells, or they can get out into the soil.
How do they get out? Does the host cell just burst?
Sometimes it might disintegrate, yeah. But other times, the zoosporangium forms an exit tube. It's like a specialized escape hatch. Part of the sporangium doesn't turn into spores, stays packed with enzymes, softens the host wall, and lets the secondary zoospores swim out.
Clever. So these secondary zoospores are now out in the soil, or maybe moving within the plant.
Right. If they're in the soil, they can infect new root hairs, just like the primary ones did. Once inside, they develop into the secondary or sporogenic plasmodium. This is the phase often linked to the major disease symptoms.
Ah, so these are the ones causing the serious hypertrophy and hyperplasia in the main root tissues, like in clubroot.
That seems to be the case. They get established in the cortex, the vascular tissues, and trigger that massive cell growth and division. As the infection progresses, you find more and more of these secondary plasmodia scattered through the distorted roots.
And these secondary plasmodia eventually make the resting spores again to complete the cycle.
Precisely. They undergo cleavage to form the thick-walled resting spores. Often, these are produced in clusters called sporosori. The shape varies, like in Spongospora, the potato scab one. They form spongy-looking masses inside the tuber.
Spongy, hence Spongospora.
Makes sense, right. In Plasmodiophora, though, the clubroot one, the resting spores might be more loosely associated or even free. Then, host dies, decays, spores are released, cycle starts over.
Wow, that's intricate. But you mentioned unanswered questions about the nuclear cycle. Meiosis.
Yeah, that's a big one. We know meiosis happens just before resting spore formation. You can see structures called synaptonomal complexes, which are hallmarks of meiosis. But when exactly plasmogamy, cytoplasm fusion, and karyogamy, nuclear fusion, happen, it's still surprisingly murky.
So we don't know for sure when the diploid stage begins.
Not definitively. There were older ideas, like for P. brassicae, suggesting secondary zoospores fuse in pairs that would be plasmogamy, and then nuclei fuse later in the secondary plasmodium. But the evidence, well, it's not totally solid.
Science is rarely completely settled, is it?
Never. And what's really interesting is that even the generalized life cycle we just described, recent work is questioning parts of it. Some studies suggest that maybe by the time secondary zoospores are even formed, the inner tissues of the plant root are already infected by amoebae or small plasmodia that came directly from the primary zoospore infection.
Wait, so the primary infection might spread deeper, faster than we thought?
Possibly. And this leads to a different idea. Maybe those secondary zoospores aren't strictly needed to form the secondary plasmodia in the deeper tissues.
So what would their main role be?
It's been proposed that maybe these little amoeboid stages derived from the primary infection are actually where plasmogamy happens, and the secondary zoospores. Maybe their main job is rapid, localized reinfection, like a microcyclic stage, to just quickly amplify the infection in the immediate area.
Fascinating. It shows how our understanding keeps shifting as we get better tools and observation.
Exactly. It's a dynamic field.
So with all this weirdness, the internal plasmodia, the cruciform division, the life cycle question, where do these things actually fit taxonomically?
Well, the phylum Plasmodiophoromycota itself is considered a solid group, what we call monophyletic. They definitely belong together, largely thanks to that unique cruciform division signature.
And their relatives are still fungus-like things.
Actually, no. This is maybe the biggest surprise. Based on molecular data, like their ribosomal DNA sequences, their ciliate relatives seem to be ciliate protists.
Ciliates, like paramecium. Things with cilia, that seems really different.
It does, doesn't it? Ciliates are known for complex cells, using cilia to move and eat. These guys are internal parasites with flagellated zoospores. It's a reminder of how evolution throws curveballs and how diverse life is at the microscopic level. Appearances aren't everything.
So classification-wise, it's pretty simple then.
Relatively. One phylum, Plasmodiophoromycota. Within that, currently just one class, Plasmodiophoromycetes, one order, Plasmodiophorales, and one family, Plasmodiophoriaceae.
Okay. And about 10 genera, 29 or so species?
Roughly, yes. And distinguishing genera like Plasmodiophora, clubroot, Spongospora, powdery scab, Polymyxa, often vectors, viruses, and cereals. It often comes down to how those resting spores are arranged. Are they single, glued together in sporosori? What shape are the sporosori?
You hinted that might be debated.
Yeah, the reliability of just using resting spore arrangement has been questioned. It's an ongoing discussion, like many things in taxonomy.
Right. Okay. Let's try and wrap our heads around this. We've gone deep into the Plasmodiophoromycota. Not fungi, but unique endoparasites.
Masters of manipulation causing those dramatic swellings and growths, hypertrophy and hyperplasia, leading to major plant diseases.
We looked inside, saw their wall-less plasmodia, different from slime molds, and that really distinctive cruciform nuclear division.
Mapped out their complex life cycle resting spores, primary and secondary zoospores with that amazing injection mechanism, primary and secondary plasmodia.
And touched on how scientists are still refining that picture, questioning stages, figuring out the details of sex and spread.
And we found their surprising evolutionary cousins, the ciliates, highlighting their unique place in the tree of life.
And underpinning all of this is their significant impact economically on crops and ecologically as pathogens and virus vectors.
So the takeaway here, maybe it's just the sheer power of the microscopic. Think about these tiny, hidden organisms operating entirely inside plant cells, yet capable of causing such huge, visible changes to plants, affecting ecosystems, impacting our food supply.
It really makes you appreciate the unseen world beneath our feet, the constant cellular battles being waged.
Doesn't it? And how much more there probably is to discover about how these intricate relationships work.
Well, thank you for joining us on this fascinating journey into the world of Plasmodiophoromycota. We hope this deep dive gave you a valuable shortcut to understanding these complex organisms.
Keep asking questions, keep exploring. There's always more beneath the surface, even in what seems like a settled chapter of biology.
Thanks for tuning into the deep dive and for being part of the Last Minute Lecture family.